Okay, so my name is Hernando Martinez and I have been donating my body to science since 2 years already when I started at Heidelberg. And for those of you who don't know, doing a PhD can be a pretty lonely job because the majority of the time you are either in a very dark room trying to make your microscope work, or you are in a very tiny corner of your desk with the only company of bacteria or cells, which don't talk very much. So at the end what you do is you end up talking to yourself so much that you start looking like Gollum. So I mean, for me to avoid having this aspect in 2 years, I want to thank Vasily very much for broadening my horizons on alternative careers. And I think I'm going to do this more often, like pretending that I know what I'm talking about in exchange for free beer. And I think I have to pay for the beer myself. So the title of my talk is Brains, 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 and that's exactly what I'm going to talk about. As I have limited time, what I decided to do is what gives you a hint about the current neurobiology techniques that scientists are employing to find out how these wonderful machines that some people have inside their heads work. So the nervous system was already known as a functional unit or as a whole more than 2,000 years ago. And it wasn't until Cajal discovery of the neuron where our way of looking at the brains changed remarkably. I mean, I think it's fair to say that Cajal he made the most important breakthrough in neurobiology in the entire history, because he changed the way we look at our brains. And the way we look at our brains nowadays is that we think about cells like very tiny computers. They're able to compute many different things, and they are also connected to each other in very different ways, and they modulate each other's activity to increase this computational power. So for scientists to explain how brains work, they need to create maps. They need to create concepts of how this machine can work, and they can understand the different pieces and how these different pieces work together. So for that, first of all, we have to see what the brains are. We have to see what is inside the brain. And I just want to show you this video. This is a mouse brain, and as you can see, it's pretty opaque. But if you remove the fat and replace it with a transparent polymer, you can really look through the brain. And in the computer, after you have taken different images that come of these layers of the brain, you can travel to the brain virtually. And you can know what is inside the brain. You can image the entire brains, even human brains, and you can stain for different neurons, like excitatory neurons. And now we are looking at the hippocampus, which is the region of the brain that creates the memories. And it's also the region that tells you how to move or controls the spatial navigation. And all these cells that you see in green are excitatory cells. These cells right here, they project to the cortex, which is the part of the brain that produces the consciousness, and they tell what is going on in the hippocampus to what we know. So this is pretty cool, and it creates very nice desktop images. But that technique gives you a little bit about, tells you how the big wires are connected, but doesn't tell you what is happening exactly in the dendrites and the axons and the connections of the neurons. This is how the neural tissue looks like. All these are extensions of the membranes of the neurons and their connections to each other. So what scientists do is they employ electron microscopy, which tells you with with a high amount of detail what is happening in the cells. And this is a very tiny piece of the mouse brain, and this is a really tiny piece of a neuron. And as you can see, there are so many connections of other neurons that the scientists can tell where these connections come from. But this is a tremendous amount of work, and this is just started to be explored. But also what you want, these techniques that I mentioned previously tells you how are the roads where information flows through. But the brains, the good thing about the brains is that they are very dynamic, and what you want to know is how the information flows through these roads. So for that you have to look at the brain when it's active. And for that, there was a very nice microscope developed here in EMBL, and together with a very cool protein that is present in the cells that the scientists can put inside the neurons. And this protein, the cool thing about it is that it changes color whenever the neuron is firing. So you can do movies like this. This is the brain of a fish, and we are looking at neural activity in life. And this is the entire brain of the fish, and it's being imaged, and scientists can tell which neuron is talking to which neuron. For example, this area here is controlling the movement of the tail. The fish is fixed in agar, so it cannot move, but it's trying to escape. And then you can tell because of this movement. And when we know more about the brain, we will be able to tell if the fish is thinking about a female fish or something like that. But this is not there yet. But it will be a pretty cool PhD. So all this, looking at the brains and observing what nature can show to us, can make scientists look for theories. But we don't like only that. We want to test these theories. So for that, you need to manipulate nature. And neurobiologists do that also using light. They can introduce another very special protein that binds into the membranes of the cell, and whenever you shine light into that neuron, the protein opens, allows the ions to go through, and this neuron is activated. So what they did to these poor mice is that they put this protein inside the cells that tell the mice to turn left. And whenever they shine light on his head, that you will see by this wire here, the animal will start walking in circles. You see, now it's fine, and now you see a blue spot here, now. And then the animal starts walking in circles. And it will continue walking in circles until the researcher turns off the light. And this is not only for movements, or not only for mice. You can also manipulate the activity of worms with the light, and you can tell a fly when to fly, for example, like now. And this application has a lot of possibilities. For example, what they have done recently is that they have put a button in the cage of the mice, and this mice has this protein inside the dopamine neurons. The dopaminergic neurons are the ones that produce reward to the animal. And what happens is that when the mice pushes the button, he gets light into the brain. So these neurons release dopamine, and this produces a reward mechanism in the brain. So what happens is that the mice forget about feeding, forget about sex, forget about everything. They only push the button because they like it a lot. I know what you're thinking. When are the clinical trials starting? Not yet, but it will be a critical thing. So what we do in the lab is that we are interested in knowing how the brain came to be as it is. So we study evolution, and we study key events in evolution and that are very relevant to the nervous system. We work with these three animals, and I'm going to focus on this worm, this marine worm, which is called Platineres, because it's the one I work with. So you will see now this movie of how they reproduce. So yeah guys, this is form. This is the female worm, and this is the male worm. And you will see now, when you put them together, they start dancing around each other, and you will see that the male is gonna flower now all the sperm that he has. And the poor female now is gonna throw the eggs away. You can see the eggs right there. And then fertilization occurs in the water, and then they die. So one day later, all these little balls have been fertilized, and now they are composed of 2,000 cells, and they swim around the water, and they compose the plankton in the sea. 4 days later, they look like this because they have elongated, and now they have legs, which they can use to move, and the whole process takes about 3 months to happen. And this is I'm going to show you some movies about this guy, because what is pretty cool and what we can do now in the lab is that this guy doesn't have so many cells, like vertebrates, which has billions of cells, so it's very difficult to characterize them. What we can do with this guy is ask for every single cell what are the synaptic partners, so which other neurons that cell interacts, and what is the plethora of genes that this cell is expressing. So we can investigate this animal both at the single cell level and at the system level. So for example, what we can do is that we can do connectomics. All these small circles that you see are cells. This is the mouth of the animal, and we are traveling from the head to the tail, cutting the animal like loaves of bread. And then these are the axons, which is like the spinal cord of the animal. And here are the connections that the animal used to move, for example. What we can also do is tell which are the genes that are expressed in every part of the animal. We can also visualize the neural activity in these guys. And we can also manipulate this activity. I can tell this guy when to stop swimming and when to continue swimming, as I shine the light. So I'm approaching the end of my talk, and if you are taking any take-home message, I would like you to know that this is all these techniques that we can employ in so many animals are gonna drive us through a world of surprises that will lead us to know ourselves a little bit better. So these days are truly remarkable days for neurobiology, and I hope that with my talk you got a hint of it. So thank you very much for your attention.